Thank you so much, Hugues, for this invitation and the opportunity to present some data, some new data, some unpublished data we are getting right now on these topics, on these specific topics, revisiting chemotherapy. As you know, relapses are the major barrier for cancer treatment, and they are due to the regrowth of drug-resistant cells that will repopulate and regenerate the tumor after initial clinical benefits. Drug resistance arises from genetic and non-genetic mechanisms and due through the selective pressures imposed by chemotherapy. And furthermore, mechanism underlying drug persistence that coexist in minimal residual disease that remain after an effective anti-cancer therapy might be implicated in initiating relapses in cancer. So in this specific context, 15 years ago, we started to work on AML, on acute leukemia, as a therapy-resistant cancer model, using a PDX, patient-derived xenograft model, to fully understand the biology of relapse-initiating cell or drug persisters enriched in minimal residual disease in vivo. So for 40 years, or mostly 50 years, the chemotherapy used in clinics for those patients at the induction phase is a combination of antimetabolite cytarabine and anthracycline idarubicin or daunorubicin. As you heard from this morning, this kind of genotoxic will inhibit DNA synthesis, and especially DNA polymerase for cytarabine and topoisomerase isomerase 2 for idarubicin. So based on this mechanism of action, those combinations will kill cycling proliferative cell and spare non-cycling cells such as cancer stem cell. So we, based on this also mechanism of action, several studies have shown that AML stem cells are responsible for drug resistance and relapse in this disease, and also when you look at different features of the cancer stem cell in this disease, they are associated with a poor prognosis. So based on the literature and on this mechanism of action, our initial question was to investigate whether residual disease in vivo in PDX is enriched in quiescent immature cancer stem cell. So we did a full analysis of leukemic stem cell biology at the MRD with a FACS analysis and also RNA-seq. And in any feature we have tested of leukemic stem cell, we never observe an enrichment in leukemic stem cell at the MRD. It can be a CD34 positive, CD38 negative phenotype, quiescence, stem cell genes, or even if in vivo limiting deletion analysis to assess the leukemic initiating cell frequency. In your right, we do not observe significant change on leukemic stem cell, suggesting this intensive chemotherapy, in fact, equally kill non-cycling and cycling AML cell, non-leukemic stem cell and leukemic stem cell. So, and in fact, when you discuss with clinicians and when you are getting back to the in vivo data, you realize 95% of the AML cell at diagnosis or before treatment in PDX are non-cycling proliferative cell in G0, G1. However, in clinics and in PDX, as you can see on the right, you have a huge debulking efficacy of this intensive chemotherapy between 1 to 3 log of total cell tumor burden reduction, highlighting the fact maybe there is others mechanism of action in addition of the DNA synthesis inhibition. So we and others have shown a couple of years ago, in fact, minimal residual disease is enriched in senescence-like resilient cell with a latent state and a stress and inflammatory response phenotype, suggesting the induction during the treatment and at the minimal residual disease of an adaptive response to stress. And we end up with this big observation of minimal residual disease enriched in relapse-initiating cell with an increase in mitochondrial oxidative metabolism, so-called high OXPHOS. Now, you have an increase in mitochondrial respiration, ATP production, an increase in TCA cycle intermediate, and in ROS content in persisters. So we know now it's this high OXPHOS phenotype of persisters is a consequence of an increase in respiratory substrate utilization, especially fatty acid fatty acid beta oxidation. A couple of papers I've mentioned also the glucose oxidation and also the branch-chain amino acid oxidation to support this high OXPHOS and to highlight the catabolic flexibility of using a respiratory substrate in drug persisters. It's also the consequence of this high OXPHOS phenotype and an increase in mitochondrial metabolic activity is the consequence of an increase in mitochondrial number, resulting from an increase in mitochondrial biogenesis and also by mitochondrial transfer from bone marrow stromal cell to ML blasts. Associated to this increase in biogenesis, we have current evidence that mitochondrial iron content and also heme and iron sulfur cluster biosynthesis is activated in this drug persister to support this mitochondrial machinery. And lastly, we published a couple of years ago, in addition of this increase in mitochondrial number, we have an increase in mitochondrial ER, endoplasmic reticulum contact site. And the function of this MERCs is to support the influx of calcium from ER to mitochondria. And in fact, we are observing an increase in mitochondrial calcium as a cofactor of calcium-sensitive TCA cycle and NADH dehydrogenase that will support the NADH availability. And NADH is a proton-electron donor of the electron transfer change complex 1. Associated to this increase in MERCs, you have a change in location of BCL2 from cytosol to mitochondria. And to associate to this increase in mitochondrial translocation of BCL2, you have an inhibition of the mitochondrial permeabilization transition pore opening, supporting these drug persisters and drug resistance. So, consistent to this BCL2 dependency, we use BCL2 inhibitor venetoclax, as used in the clinics, in fact, in association with low-dose cytarabine. As you can see in these 7 different PDX, in orange, the combination is better than the chemotherapy alone or plus placebo in all of the case. And that is consistent to the observation we did in presence of venetoclax. You block the activation of the high OXPHOS activity in orange compared to blue, as you can see with mitochondrial respiration or oxygen consumption rate, the flux of ATP, or the TCA cycle metabolite content. And will completely abrogate the stimulation of this high OXPHOS by RC. So more broadly, when you target selectively any of the electron transfer change complexes with a selective inhibitor or indirect inhibitor, you lead to the increase in sensitivity to cytarabine in vitro and in vivo. So we next ask the question how we can explain this high OXPHOS phenotype. And in fact, we realize this high OXPHOS phenotype reflects indeed a mitochondrial stress response induced by ATF4-driven transcriptional program and adenosine CD39 pathway upon RRC treatment, and leading to the mitochondrial recovery by stimulating the fatty acid oxidation, antioxidant defense, and also the mitochondrial biogenesis. And in fact, if you target with a small molecule inhibitor, monoclonal antibodies, or shRNA, this pathway of adenosine CD39 protein kinase signaling pathway or ATF4 stress pathway, you block the mitochondrial stress response and you block the mitochondrial recovery that lead to a decrease in mitochondrial activity and a synergy to cytarabine. This is in vivo with a POM1 inhibitor, CD39 inhibitor. We realize also its current work we have in revision using the second drug, I tell you at the beginning in the introduction, idarubicin. In fact, you are synergizing RRC effect by the same mechanism of blockage of the mitochondrial recovery through the ATF4 inhibition, as you can see here. And in fact, we are currently proposing a novel mechanism of synergy between these two drugs to explain the debulking activity I mentioned at the beginning as well. So next, we are currently investigating the evolution of this mitochondrial adaptive stress response and the OXPHOS state in vivo. So we did longitudinal analysis on mitochondrial biology just after the treatment, 1 week after, and 3 weeks after. By FACS analysis, multiparametric mito and ROS flow cytometry, and also RNA-seq analysis. As you can see here, in a 10 different ML PDX model, we have a significant reduction of the total cell tumor burden on your left. And when we look at the different mechanism of resistance, MRD, RRC up or relapse up, in fact, we have two different patterns, two different transcriptional patterns. So we decided to look at what kind of, in these two different response, when you are up during the treatment and then down at relapse, or down during the treatment and then up at relapse, what we are getting. But in fact, when you look at the early response, this correlates with inflammatory response again. And as you can see here, the inflammatory response or the different gene signature related to senescence or inflammatory are transient. And in a late response, in fact, we have an enrichment in signature related to leukemic stem cell. And we are checking a lot of different signatures, and every time we are getting this profile. So next, we look at our high OXPHOS signature at the transcriptional level. And as you can see, again, fatty acid oxidation, high OXPHOS, ROS, gene signature, the evolution, it's transient again after treatment. So we decided to confirm that more functionally using flow cytometry. We look at mitochondrial state, FAO state, and also ROS state by flow cytometry, and corresponding to the transcriptional program and the kinetics of this transcriptional program, we observe a transient increase of mitochondrial mass, mitochondrial potential, and mitochondrial calcium. We observe also a transient increase in CD36, it's a transporter of fatty acid to support beta oxidation, and also by CNIT, we observe a transient increase of fatty acid, amino acid capacity. And again, we got exactly the same pattern with the total ROS and mitochondrial ROS. So we decided to look at CD39 mitochondrial stress response and ATF4 as well. As you can see here, in parallel of what we observe with high oxos gene signature, we have exactly the same transcriptional behavior, and we confirm with CD39 expression, suggesting the selection of a transient selection of a specific cell population. So, based on these two transcriptional profiles, we have two different, in our opinion, of response. An early response related to the mitochondrial stress response, and a late response associated to the leukemic stem cell-like resistance response. So we decided to investigate this mitochondrial oxostate and the cellular leukemic stem cell composition of the MRD at the single cell level to identify if it's the same cell population or different cell population supporting these two transcriptional programs. And this work is done by a very talented PhD student currently in our lab, and doing a longitudinal multi-omics analysis of mitochondrial biology and cellular heterogeneity of the MRD at different time points during the treatment, after the treatment, at the minimal residual disease, and after a relapse with a single-cell RNA-seq and different other multi-omics. But today I will present only the single-cell RNA-seq. But we will try to integrate all of that to make a better sense of what I will present. It's very preliminary, but I think it's an overall mechanism of multi-drug tolerance mechanism. So we did 10x genomics before and after treatment, during and after treatment, to assess the transcriptional proximity of the different cells. We did 6 different PDX models, and on the right you have the SERAT integration with 9 different transcriptional distinct drug resistance cell clusters in vivo, and we have 6 time points. And we have sequence almost 100,000 human ML cells on this, from this PDX. So first, we decided to look at, as we did with the bulk and the gene signature, the cellular behavior of these 9 different clusters on the left. As you can see, some are up at the minimal residual disease, and some are up at the relapse. So we decided to cluster and to reduce, in fact, the cellular heterogeneity from 9 persister cell cluster to 3 major drug-tolerant persisting cell population with 3 different cellular behavior in vivo. So we first look at our different leukemic stem cell gene signature, and as you can observe on this UMAP, you have different subgroup of, based on the gene signature of leukemic stem cell and progenitor cell. So the cluster 2 enriched at relapse, it enriched in LSPC cycling, while the cluster 3 enriched at the minimal residual disease is associated to a gene signature related to inflammatory leukemic stem cell. The cluster 1, that is more stable over time, is associated to prime or quiescent leukemic stem cell gene signature. So we look at different mechanisms of resistance. Genes upregulate in low responder to RRC. RRC up, and also relapse up. As you can see here, these 3 different cell populations, it's associated differently to these different mechanisms of resistance. And we try to correlate with different metabolic pathway, and today I'm presenting 3 different high OXPHOS related to the cell population 2, the HIF target genes signature with a cluster 3, and the fatty acid beta oxidation with mostly CP1 and also CP2 as well. We did also an enrichment analysis, and as you can see here, the cluster 2, the population 2 is related to cell cycle, cell division, and also some DNA repair pathway. The cluster, the population 3 is related to response to stress, inflammatory stress, defense response as well. And the population 1 associated to the quiescent leukemic stem cell is mostly dependent on translation and also on ribogenesis. We then look at the most upregulated genes in each of these cell population, and especially we want to sort those cell population, and we look at the receptor as a cell surface marker. As you can see here, we were able to identify some key cell surface marker on the 3 different populations. They are not overlapping at all. And the cluster 3 was associated to especially CD39 and CD36. So we decided to validate this cell population by doing cell sorting at the different time point based on CD39 expression, and then to do limiting deletion analysis, LTCIC analysis, FACS analysis, and also RNA-seq, and some drug OXPHOS inhibitor screening to verify if CD39 high is associated to this inflammatory leukemic cell, stem cell or progenitor, if it's high OXPHOS, etc., etc. So, as you can see here, we did 5 different PDX, plus and minus RRC, and today I'm presenting just before treatment and after treatment, and we'll have the relapse shorter. In these 5 PDX, the first graph is single sample GSEA, comparing the score of high OXPHOS gene signature in high versus low CD39 before and after treatment. As you can see here, you observe a significant increase in high OXPHOS score, mostly in diagnosis. We have a trend at MRD. And we were able, for 1 PDX, to do oxygen consumption with seahorse, and you observe exactly the same pattern with the functional assay. We did also FACS analysis of mitochondria state with mitochondria mass, mitochondria membrane potential, and mitochondria calcium. Every time you have an increase in between the 2 cell population in this parameter, but you observe in both cell fractions the same trend, the same transient trend. Then we look at the inflammation. Again, in high OXPHOS, in high CD39 cell population, we observe an enrichment in gene signature related to senescence-like, resilience, ROS, and also the specific leukemic stem cell from SATO study. It's an inflammatory leukemic stem cell. Then we look at by flow cytometry, ROS, mitochondria ROS, and total ROS, and as well the size and the granularity of the cell. It's one of the readout of the senescence like. As you can observe, in high CD39, you have an increase in size in mitochondria and total ROS content. But the trend is the same in high and versus low as well. It's transient again, a transient phenomenon. And finally, we check leukemic stem cell state with different signature before and after treatment. As you can observe on the right, all of the gene signature are downregulated or depleted in CD39 high fraction, as we observed previously at the bulk level. And when we did limiting dish analysis in 4 different PDX, after sorting and doing a limiting dish analysis in secondary recipient, the high CD39 fraction in both before and after RRC are lower in leukemic initiating cell frequency, suggesting there is some leukemic stem cell potential, but they are lower than in CD39 low cell subpopulation. And finally, we check the most upregulated genes in CD39 of high fraction compared to low fraction before and after treatment. As you can observe, CD39 is up, so we did a good job, and Emily did obviously a good job. And also we observe SOT2 and MPT1 a significant increase in both RRC and vehicle-treated high CD39 fraction compared to low fraction. And again, some marker of the leukemic stem cells, such as CD39, was completely decreased and downregulate in high CD39 fraction. So we confirm that, or we are currently confirming that ex vivo with ML patient sample. Again, we observe this kind of increase in mitochondrial state, a decrease in leukemic stem cell phenotype between high CD39 and low CD39. So as a take-home message, I have 3 slides, so I will be finished. The bulk minimal residual disease is enriched in drug resistance persisting cell with an increase in oxidative OX-phos metabolism, an increase in mitochondrial biogenesis and fatty acid oxidation, and also a mitochondrial BCL2 dependence as a consequence of an adaptive and transient response to chemotherapy-induced mitochondrial oxidative stress, so-called mitohormesis, in TF4 and adenosine dependent protein kinase pathway dependent manner. The inflammatory senescence-like state can facilitate AML cell survival to genotoxic by allowing cell to persist in a latent state while retaining some leukemic repopulating activity. And in our context, mitochondrial stress response can connect respiration, inflammation, and apoptosis as an evolutionary interplay between mitochondrial metabolism and drug persistence within minimal residual disease. At the single cell level, we observe a multiple drug tolerant state that can be present at diagnosis, transiently coexisting at minimal residual disease, and differentially co-evolve upon disease progression after treatment. MRD and or relapse-enriched drug persisting cell population, DPCP, so-called now by us, highlight an apparent transcriptional and cellular uncoupling between OX-phos and fatty acid oxidation capacity and inflammatory senescence-like versus leukemic stem cell-like state. So it's still in progress. We will better characterize this different cell population, and we'll try to confirm by genetics and also pharmacologic approach to now target not only the OX-phos or the FAO, but by doing triplet therapies or quadruplet therapies to try to target the different drug tolerance mechanism. So the current question we have is to identify the earliest signal or event that drive mitochondrial adaptation and selection of drug persistent cell population in vivo. We have some evidence, still preliminary, of the role of mitochondrial ROS, NAD plus, and NADH redox, and also electron transfer change complex 1 and 2 through the reverse electron transfer flow. and also lipid signalization and inflammation. We would like to trace mitochondrial-dependent cell population to investigate the coevolutionary, not only the metabolic, but also the epigenetic and the genetic mechanism of drug persistence, and to identify the cell cluster as the origin of the relapse in vivo. We are thinking about the role of CD39 TF4-dependent leukemic progenitor cell subpopulation. And also, we would like to know whether metabolic transition or this mitochondria adaptation or mitohormetic process occur in drug persistence of other type of drugs used in clinics, in idarubicin alone. In CPX, it's a new genotoxic use. It's a liposomal daunorubicin plus RRC, venetoclax combination, and diverse oxos inhibitor in vivo. And for the clinical part, we think we have to develop a combinatory metabolic precision medicine targeting or preventing mitochondrial adaptation and metabolic evolution with an anti-AML cocktail alternating chemotherapy or BCL2 inhibitor in association with OXPHOS inhibitor and or personalized diet based on relevant mitochondrial biomarker. We are developing MitoScore, and also based on the minimal residual disease cell composition. More specifically, we would like to develop therapies that selectively target CD39, BCL2, NMPT1, CPT1 of the drug persisters cell population to know if we can prolong my survival with this kind of triplet therapy that also will use the alternance between chemotherapy, venetoclax combination with this different inhibitor that will try to reduce mitochondrial adaptation and residual disease heterogeneity that might be delay relapse. So that was in AML. Since our work in 2017, plenty of other groups have shown that also in AML, after different drugs, the role of the changes in mitochondrial energetic metabolism and structure as a hallmark of the drug resistance, not only after intensive chemotherapy, but also targeting therapies. More specifically, we think about the central role of the adaptation in mitochondrial dynamics and oxos flexibility in lipid metabolism during treatment, driving residual disease and drug tolerance in AML. Now you have plenty of also study about the role of mitochondrial metabolism associated with drug resistance and persistence in other blood cancer, but also in several therapy-resistant solid tumor, including melanoma, PDAC, breast cancer, sarcoma, and also at some metastatic grade. So I would like to thank my team, the former member and the current member, especially Emeline Bouet, that doing the second part of the work. And our collaborator, we have a very intense and close collaboration with clinicians and also with people running metabolomics and fluxomics and mitochondria core facility. Some collaborators in France and the US, especially Jérôme Tamburini, Yves Colette, and Martin Carroll and Sarah Scotland at Penn. And our findings, and I will leave you with this sentence I'm convinced about. Thank you so much for your attention.